Welcome to the fascinating world of biological data generation. Imagine you have a massive billion-page instruction manual for building a human being, but it has been shredded into millions of tiny pieces. Before we can analyze biological data on a computer, we first have to extract it from the messy, wet biology of a living cell. This process of turning physical DNA, RNA, and proteins into digital text is the very foundation of computational biology. Let us dive in and see how this is done. Now that we understand our goal, let us look at our first technology, gene sequencing. Why is this concept so absolutely critical to our modern world? Without gene sequencing, the entire field of bioinformatics would simply not exist. Modern techniques, known as high-throughput sequencing, do not read the whole genome from start to finish. Instead, they randomly break the DNA into millions of short fragments, read them all simultaneously, and generate massive digital text files containing these short sequences called reads. Computational biologists then use complex algorithms to align and assemble these short reads by finding overlapping regions, ultimately reconstructing the original, complete genome. This process of reading fragments leads us to an important mathematical question. How do we know we have read enough? We use the sequencing coverage formula. Conceptually, this tells us how many times, on average, a single base in the genome has been read by the machine. In this equation, C represents the sequencing coverage. L stands for the average length of each short DNA read, measured in base pairs. N represents the total number of reads generated. Finally, G represents the total length of the target genome. If you increase the number of reads, N, your coverage C increases, meaning you have much more confidence in your final assembled sequence. Before we move on, let us see this with a concrete example. Suppose you are sequencing a small bacterial genome with a total length of 4 million base pairs. Your machine generates 200,000 reads, each with an average length of 100 base pairs. We want to find the average sequencing coverage to know if we have enough data. We multiply the read length of 100 by the 200,000 reads to get 20 million total sequenced bases. Dividing this by the 4 million base pair genome size gives us a final coverage of 5. This means every letter was read 5 times on average. Now that we have calculated coverage, let us try a slightly harder, real-world problem involving the human genome. The human genome is roughly 3 billion base pairs. If we want a high-quality coverage of 30 and our reads are 150 base pairs long, how many total reads must the machine generate? We rearrange our formula to solve for N, multiplying coverage by genome size and dividing by read length. We plug in 30 for coverage, 3 billion for genome size, and 150 for read length. The numerator becomes 90 billion. Dividing by 150 gives us 600 million reads. Generating this much data takes immense computational power. Having calculated how many reads we need, let us visualize what this actually looks like. Here you are seeing a diagram of DNA read alignment coverage. At the top, the long horizontal line represents the complete reference genome. Below it, you can see many short, overlapping horizontal line segments. These represent the individual short DNA reads generated by the sequencing machine. The vertical dashed lines show how these short reads align to specific positions on the reference genome. Notice how multiple short reads stack up under the same region. The number of overlapping reads at any specific vertical slice is exactly the coverage we just calculated. While sequencing gives us the static blueprint, it does not tell us what the cell is actively doing. This brings us to microarrays and gene expression data. Why do we need to know which genes are turned on or off? Because every cell in your body has the exact same DNA, yet a heart cell behaves very differently from a brain cell. A DNA microarray is like a giant switchboard. It is a chip printed with thousands of microscopic spots, each representing a single gene. When a cell is active, it produces RNA. We extract this RNA, label it with fluorescent dyes, and wash it over the chip. A laser scanner measures the brightness to determine how active each gene is. To make sense of this fluorescence data, we need a way to quantify the change in gene activity. We use the log-base 2 fold change formula. Conceptually, this metric expresses how much a gene's activity has increased or decreased compared to a baseline. In this formula, L stands for the log-base 2 fold change. E sub t represents the fluorescence intensity, or expression level, in the treatment or diseased sample. E sub c represents the expression level in the normal, healthy control sample. We divide the treatment expression by the control expression to get a raw ratio, and then wrap it in a base 2 logarithm. This makes the data symmetrical for both increases and decreases in expression. Let us apply this formula to a specific gene suspected to be involved in lung cancer. In our healthy control tissue, the expression level is measured at 400 units of fluorescence. In the cancerous tissue, it spikes to 3,200 units. We want to quantify exactly how much this gene is overexpressed. We substitute our values, placing 3,200 in the numerator and 400 in the denominator. Dividing these gives a raw fold change of 8, meaning the cancer cell produces 8 times as much RNA. Finally, the base 2 logarithm of 8 is 3. This gene is upregulated by a log fold change of 3, making it a prime target for new drugs. We have looked at DNA and RNA, but what about the actual physical workers inside the cell? Now let us explore how we generate data about proteins using mass spectrometry. While DNA provides the instructions, proteins are the actual molecules that build tissues and fight infections. Sometimes a gene is normal, but the resulting protein is faulty. Mass spectrometry allows us to directly measure which proteins are present. It works by digesting proteins into smaller pieces called peptides, vaporizing them, and giving them an electrical charge. The machine then uses electric and magnetic fields to accelerate these ions, measuring exactly how long it takes them to hit a detector to determine their precise mass. To identify these flying peptide fragments, the machine relies on a specific physical relationship. This is the fundamental equation for the mass-to-charge ratio. Conceptually, it calculates the effective mass of the ionized particle as it travels through the spectrometer. Here, R denotes the measured mass-to-charge ratio. M represents the true molecular mass of the neutral peptide fragment before ionization, measured in Daltons. The variable Z represents the integer electrical charge added to the peptide, like picking up extra protons. Finally, H represents the mass of a single proton, which is approximately 1 Dalton. If the charge Z increases, the measured ratio R will decrease, shifting the signal on the resulting mass spectrum graph. Let us see how a computational algorithm interprets this raw mass spectrometry data. Suppose a specific peptide fragment has a true neutral mass of 1046.5 daltons. During ionization, it picks up 2 protons, giving it a charge of 2. Assuming a proton mass of 1.0 daltons, what ratio will the detector record? We plug our values into the formula. First, we calculate the total mass of the added protons by multiplying the charge of 2 by the proton mass of 1, giving 2 daltons. Adding this to the neutral mass gives a total ionized mass of 1048.5 daltons. Dividing this by the charge of 2 gives a final mass-to-charge ratio of 524.25. We have covered a tremendous amount of ground today, so let us review our key takeaways. We started with gene sequencing, which generates the foundational digital blueprint of an organism's DNA. We then explored microarrays, which bridge the gap between static DNA and dynamic cellular behavior by measuring active gene expression. Finally, we looked at mass spectrometry, a brilliant physical technique that identifies the actual proteins operating within the cell. Together, these 3 data generation techniques provide the raw material that makes all computational biology possible. They are the eyes and ears that allow computers to perceive the biological world. You now understand the foundation of biological data.